Okay, so welcome to this next video in the playlist on voltage-gated ion channels. In this video what we're going to discuss is a channel that is called often the BK channel for big potassium current, basically, large conductance potassium channel. But actually it is voltage-gated, but more so it's calcium-gated. Okay, so what we're going to discuss in this video is the BK channel, so I'll put that as the title, BK channel, and it's basically a large conductance, this is its full name, a large conductance, which is what the B is for, big conductance if you like, and it's voltage and calcium gated, but calcium is the important one here, voltage and calcium gated potassium channel. Okay, and it's got a very, very similar structure to the voltage gated potassium channels that we have considered. So basically we'll start off with its structure, we'll then talk about its function, and then we'll talk about a few little nice examples of its function basically. Okay, so let's start with its structure then. So it's constructed from 4 alpha subunits, just like in the case of the voltage-gated potassium channels. So it's constructed from 4 alpha subunits, and basically there is only one gene coding for this alpha subunit. So that's very nice, isn't it? 4 alpha subunits, and there's one gene that codes for this this alpha subunit that makes up the channel. And that gene is called the KCA1 gene. So this is the gene here, KCA1. So potassium channel, which is calcium gated, and it's the first type. And this first type, because there are other calcium gated potassium channels, which we'll discuss in later videos. In fact, let's just briefly talk about calcium gated potassium channels now. So of the potassium channels, there are absolutely loads of potassium channels. Of the ones that are grouped into the 6TM P group, so remember those are the ones where the alpha subunit had 6 membrane-spanning alpha helices and then a P loop like so. So the channels which were made out of alpha subunits that looked like this. And you put 4 of these together to make a functional channel. So of these sort of potassium channels, there are 2 main subtypes. There are the KV channels, i.e. the voltage-gated ones. So there are loads of genes which code for structures like this, which aggregate together to, you get 4 of them to aggregate together, and they form voltage-gated potassium channels. On the other hand, there are also the KCAs, which are calcium-gated potassium channels. So there are genes which code for a structure like this, again called the alpha subunit, which joins together with 4 other alpha subunits to make a structure that looks semi-like, how should I draw this? So 4 cylinders effectively that surround a pore like so. And we will study the structure of these channels in more detail when we study how you actually move ions through them. Okay, but basically the gene codes for a single one of these cylinders, which as far as the polypeptide structure looks like this, and all of these obviously coil up together to make this cylinder structure, and then 4 of those aggregate together to make a potassium channel. Basically there are absolutely loads of genes for polypeptides that look like this. Some of them will make voltage-gated potassium channels, and some of them will make KCAs, calcium-gated potassium channels. We know that the voltage-gated potassium channels are then subdivided further into KV1 all the way down to KV12s, and then within each of those subclasses there are loads of genes in there. For instance, KV1 has KV1.1 all the way down to KV1.8. So there are absolutely loads of genes, and in total I think there are 40 known genes which code for alpha subunits which aggregate together to make voltage-gated potassium channels. Okay, KCAs, there are less basically that are known. So there's KCA1, and there's only one KCA1, and it goes all the way down to KCA5, and other ones of these subunits here, other ones of these subclasses here, so the KCA1, KCA2, KCA3, KCA4, KCA5, they have more genes. So for instance, KCA2, there are 3 different genes which are all classified as being KCA2s. So there's KCA2.1, KCA2.2, I'm making loads of mistakes, KCA 2.2, etc. Okay, but there is only one gene which codes for a KCA1 potassium channel. Okay, so this one gene codes for this KCA1 alpha subunit, and basically 4 of those alpha subunits are going to aggregate together to make a BK channel, basically. Okay, so let me talk about the structure of the polypeptide which is made from one of these KCA1 genes. So let's draw the KCA1 gene. So here is a piece of DNA here with the KCA1 gene on it. So here is the KCA1 gene. Right, so this codes for a polypeptide, basically. So we make a polypeptide from this, which I will draw here. So it's basically got a slightly different structure from this. When this polypeptide folds and is implanted into the membrane, it has a slightly different structure from this. It has actually 7 transmembrane spanning domains. So it's still classified as a 6-TMP, even though it actually has 7 transmembrane domains. So it's a slight variant on this, but it's very, very similar to the rest of these, so it's still kept in this same classification scheme. Okay, it's more similar to these types of subunits which make up potassium channels than it is to the other types which can make up potassium channels. Okay, so basically its structure looks like this. So if the cell membrane is here, then you have one membrane spanning alpha helix coming down here. So the amino terminus is actually extracellular in this alpha subunit of the KCA1 channel. You first have this first membrane spanning domain, which is called S0. So you don't usually have an S0. Usually in this labeling you start S1 is this first membrane spanning domain, then S2, then S3, then S4, then S5, the P loop, and then S6. In this case you have an extra 7th one, which is called S0. Then it's the same after that: S1, S2, S3, S4, S5, the P loop, and S6. Now, before you get to the carboxyl terminus, you then have 2 very big cytosolic domains on this side, which are really important. Here's the carboxyl terminus, and you have 2 regulatory cytosolic units here, and these basically are these portions of this protein that are going to sense calcium. So this is how this channel becomes calcium and voltage gated. So it's voltage gated because it's got this structure up here, which is very similar to the other voltage gated potassium channels, but it's calcium gated because it's got these 2 bits here, which are new, basically. So these are called the regulator of the conductance of potassium domain 1 and the regulator of the conductance of potassium domain 2, so RCK2. Okay, so that's what those two cytosolic domains are called, and basically both of these domains have a small portion, a small little hole where calcium binds, and these are known as calcium balls. Calcium balls, and basically you have lots of carbonyl groups from the amino acids pointing into these calcium balls. So let me remind you of the structure of a polypeptide. Okay, so firstly we'll start with an amino acid. So here is an amino acid. So you start off with the amino terminus bonded to the alpha carbon in the middle, and then you have the carboxyl group over here. Right, and then you have a hydrogen here, and then the R group, which determines which amino acid it is over here. Now, if you want to bind this to another amino acid to start making a polypeptide, then what you do is you take off this hydroxyl group, bind this carbon to a nitrogen here, and then you have a second amino acid that's going to go here. So you have CH, then another R group, which I'll call R prime, and then another carboxyl group there. Okay, so this hydroxyl group has come off here, so I'll cross that out. And basically this is another amino acid structure here with one of these hydrogens removed off there. So when you build amino acids, what you end up with is lots of these groups here, which are known as carbonyl groups. So this is a carbonyl group. Okay, and basically oxygen's electronegativity is far greater than carbon's electronegativity, meaning that the attraction of the electrons in this bond, in this double covalent bond, to the oxygen is greater than the attraction of the electrons to the carbon. So basically, both the oxygen and the carbon have nuclei. They're atoms, so they have nuclei, which contain protons. So the nuclei are positively charged. The electrons are negatively charged. They are going to be attracted to the nuclei of the carbon and the nuclei of the oxygen. Basically, the pull of those electrons towards the nuclei of the oxygen is greater than it is to the nuclei of the carbon. So the electrons actually spend most of the time closer to the oxygen atom than they are to the carbon atom. So that overall means that this structure here becomes polar, because you end up with more negative charge over here than you end up with here. So you end up with a partial negative charge over here and a partial positive charge over here. Basically, in these calcium bowls, you have loads of these carbonyl, the oxygens of these carbonyl groups sticking into the center of that calcium bowl, and that will interact with the calcium ion sitting at the center. And obviously, if this is negative charge, then the calcium will like to interact with that oxygen. So that's how you get calcium to bind there, basically. You have these carbonyl groups which it's going to be attracted to electrostatically. Okay, right, so that's pretty much the structure of a single alpha subunit. Now we need to discuss the more macroscopic structure of the entire calcium-gated potassium channel. Okay, so, oh, and actually before we do that, I just want to mention something about this subunit. So you might think we only have one gene, so you might think that you can only therefore make one type of calcium, voltage-gated calcium channel, basically, from, sorry, not a voltage-gated calcium channel, only one type of these BK channels, which is a calcium-gated potassium channel. However, that's not the case. You can get variation in the structure of this alpha subunit of the BK channel, basically. And the reason is that this KCA1 gene has a huge number of exons, basically. So if I draw on the exons, so the pink lines will mark the start of an exon and the end of an exon. So basically you have a huge number of exons in this KCA1 gene. And the portions of DNA in between are introns. So this is an intron, and this here is an exon. Basically, you do not have to use all of the exons to make a functional channel. Some of them are optional. Some of them obviously are essential. The ones, you know, that are coding for RCK1, RCK2, they're going to be pretty essential. But there are quite a few of these which are optional, basically, and you can choose whether you want to have it in the alpha subunit or whether you don't want to have it. So basically, when it comes to the stage of splicing, what's going to happen basically is this whole gene is going to be transcribed. So you're going to get a huge great piece of pre-mRNA, which will have all the introns in still. So you have the exons, introns, exon, intron, you know. So it makes an exact copy of this in the mRNA. Okay, so this is a piece of pre-mRNA. And basically, when you turn this into a piece of mature mRNA, what you're going to do is chop out all the introns. And the other thing is you can decide which of the exons you're going to keep. So you'll have to keep all of the essential ones, otherwise you're not going to make a functional alpha subunit for this BK channel. However, there are some exons which don't need to be there in order for this to be functional, and therefore you can choose whether they're going to be in there. So that gives you variation in the structure of the alpha subunit of the BK channel. Right, so okay, so you make 4 of these alpha subunits of the BK channel, and you join them together, basically. Okay, so here is the membrane of the cell, and basically you now have 4 of these alpha subunits of the BK channel, 4 of these KCA1 channels, subunits, and that overall now makes us a functional, large conductance, voltage and calcium-gated potassium channel, or a BK channel, basically. Okay, but there are auxiliary subunits as well. There is a subunit called the beta there are beta subunits that can join in on the fun, basically. Now, beta subunits, there are more genes coding for beta subunits that can join this BK channel. There are actually 3 different genes for beta subunits. So if I draw some DNA here, then what we have is 3 different genes, and they're probably not next to each other, but for the basis of the picture, I will draw the 3 different beta subunits next to each other. So let's say this is a gene for a beta subunit, this is another gene for a beta subunit, and this is a gene for the beta subunit. So there are 3 different genes coding for beta subunits: beta 1, beta 2, and beta 3. Okay, and basically they code for a polypeptide that, as far as its membrane spanning structure is concerned, looks kind of like this. Okay, so you end up with 2 membrane spanning alpha helices, and you end up with both the amino terminus and the carboxyl terminus on the cytosolic side. Okay, so basically, when you make a BK channel, you have to have the 4 alpha subunits, but you can also join on beta subunits, and you can put in 4 beta subunits. So, 1 beta subunit, how shall I mark these in? 1 beta subunit sits kind of in between each of the alpha subunits. So let's put a beta subunit there, let's put a beta subunit here, a beta subunit here, and a beta subunit there. So that's where they kind of sit, and that overall now is the large conductance voltage and calcium gated potassium channel, or the BK channel, finished. And basically these beta subunits alter the function slightly of the channel, so they can alter, the voltage gating, they can alter what threshold potential this will open at, they can alter the calcium sensitivity, and they can alter the conductance. They can also alter the inactivation kinetics of this, once it's open, how long does it remain open before it inactivates. So they alter the properties of them, basically, and we'll see a very important example of a beta subunit that does something that's clinically interesting. Okay, later. Anyway, now what I want to talk about is what is the main function of these BK channels, because we're going to see some examples of functions of BK channels, but they're not the main example. The main example of where these BK channels are and what they're doing, I want to discuss now. So basically, if this is a neuron, the axon terminal of a neuron, so let's say this is the axon terminal of a neuron. So here we have the axon, and this here is the axon terminal. Right, so basically, one of the places where you find these large conductance voltage and calcium-gated potassium channels is you find them in the axon terminals of neurons, and you find them specifically coupled to voltage-gated calcium channels. So, let me draw a voltage-gated calcium channel. Now, this is a nice bit of a revision of the structure of a voltage-gated calcium channel. So, remember, a voltage-gated calcium channel, you have the alpha subunit, which has 4 domains, 1, 2, 3, and 4. And in the case of voltage-gated calcium channels, all of those are a single polypeptide, remember. So, you've got 4 times 6 membrane-spanning domains, so 24 membrane-spanning alpha helices in 1 polypeptide. So it's all one great big polypeptide. Okay, then you have the accessory subunit, so here is a gamma subunit. Then you have the beta subunit, which sits between the first and second domains. Then you have the alpha-2 delta subunit, which sits over here. Right, so let me highlight that all in as a voltage-gated calcium channel. So this is our voltage-gated calcium channel, this. Right, okay, so specifically that be an N-type voltage-gated calcium channel, which just means that the alpha subunit, which is this main bit here with the pore through it, is coded for by the CAV 2.2 gene, basically. So N-type voltage-gated calcium channel. Right. These BK channels are found linked to these N-type voltage-gated calcium channels. Not just any old type of voltage-gated calcium channel, specifically these N-type voltage-gated calcium channel. So let me draw in a BK channel next to this voltage-gated calcium channel. So here it is. That's good. Okay, and I should put on some of those beta subunits as well, maybe. So I'll put in the green lines to represent the beta subunits. Okay, so there we go. Right, so that it represents our BK channel, or our large conductance voltage and calcium-gated potassium channel. Right, basically, now a bit of revision of what happens in the action potential. So, if this neuron fires lots and lots of action potentials in a short period of time, what is going to happen? Well, when it fires action potentials down its axon, the action potentials get to the axon terminal, and when they get to the axon terminal, they cause depolarization of the cell membrane, basically. So voltage-gated sodium channels, which I've drawn up here, here's our voltage-gated sodium channel. It doesn't get the privilege of having its actual structure drawn. This voltage-gated sodium channel, in fact, we should have drawn its actual structure, so let's try and rectify this picture. Here is the alpha subunit of the voltage-gated sodium channel, which again is a single polypeptide that makes up that entire alpha subunit, and it also has an accessory subunit, which is the beta subunit. Okay, so that's the voltage-gated sodium channel there. And the voltage-gated sodium channels open. They allow sodium to enter the cell from the outside, and that depolarizes the cell membrane to about plus 40 millivolts. Voltage-gated calcium channels, these N-type voltage-gated calcium channels, are going to open because you have depolarized the membrane, and they will allow calcium into the cell, basically. So calcium is going to come into the cell, and we know that will lead to neurotransmission, basically. The calcium is going to bind to synaptotagmin, which will lead to the fusion of the synaptic vesicle. So I'll draw a synaptic vesicle here. So it's going to lead to the release of the neurotransmitter into the synaptic cleft. So I'll draw a neurotransmitter here. Okay, right. However, if you fire action potentials very, very rapidly, loads and loads of action potentials in a minute, let's say, then what gradually happens is that the calcium inside this axon terminal is going to get higher and higher and higher, because you have to, to get the calcium back out. How do you get it back out? You have to pump it out, but that takes time. If you are firing action potentials very, very rapidly, then the rate at which these voltage-gated calcium channels are letting calcium in might it might be too big to be dealt with by the pumps, basically, i.e. the rate at which calcium is coming in might be bigger than the rate at which it's going out. That will lead to calcium building up in the axon terminal, and that's dangerous because calcium leads to problems within the cell. It can, it's one of the signals for apoptosis to begin. Right, so you need to do something to try and stop more action potentials firing and try and stop more calcium coming in. And one of the mechanisms underlying that is this BK channel. So basically, if calcium builds up, what it's going to do is it's going to activate this BK channel. It's going to activate this large conductance voltage and calcium-gated potassium channel. So calcium is going to bind to those calcium balls in the regulator of conductance of potassium 1 and regulator of conductance of potassium 2 domains. So let me just remind you, each of these alpha subunits, remember, had 7 transmembrane, membrane spanning alpha helices, and then it had 2 calcium sensitive domains down here, the regulator of conductance of potassium 1 and the regulator of conductance of potassium 2. And basically, each of those has a calcium ball. So calcium is going to bind to these regulator of conductance of potassium 1 and the regulator of conductance of potassium 2, and that's going to cause this BK channel basically to open. So when the BK channel opens, that will allow potassium to leave the cell, basically. So potassium is going to leave the cell, okay, and you're going to create another potassium. So basically, this creates a potassium current leaving the cell. And now it's time to talk about resting membrane potential across cells. So if you have a cell here, which is at resting membrane potential, negative 65 millivolts is the usual resting membrane potential for cells, then at that resting membrane potential, what usually happens is that you have a conductant, is that you have currents of sodium into the cell from outside. Because remember, you have big concentration gradients. Sodium is much higher outside the cell than it is inside the cell, and potassium is much lower outside the cell than it is within the cell. Okay, so sodium, basically, you're going to get a net movement of sodium into the cell, because both the concentration gradient and the electrical gradient are going to lead to the probability of a sodium molecule moving from the extracellular compartment to the intracellular compartment being greater than the probability that a sodium molecule will move from the intracellular to the extracellular compartment. Okay, so you're going to have a net movement of sodium into the cell. You're also, if we draw in a leaky potassium channel, and I should have said that, cells have channels in them which are open all the time, basically, which allow sodium and potassium to cross. So you have leaky sodium channels and leaky potassium channels. You also have leaky potassium channels in the cell membrane. Now, the potassium concentration is much higher within the cell than it is outside the cell. So the chance of a potassium ion hitting this channel and just happening to go through from the intracellular to the extracellular domain is much higher because there's a much higher concentration here. So they're much more likely to go this way than they are to come this way, because just, if you've got absolutely, let me give you a picture. If the potassium concentration is really high in here and really low here, what's the chance of one of these ions hitting there from this aspect? Much higher than one of these ions hitting here from this aspect. So the number that's going to go through from this side to this side is bigger than the number from this side to this side. Now, the electrical potential difference across the membrane is negative 65 millivolts. That means that the intracellular compartment is more negative than the extracellular compartment. Potassium has a positive charge and likes to be in areas of negative electrical potential. So that's going to, the potassiums will want to stay here more. So that is going to reduce the probability that a potassium ion in here will move out of here. But it doesn't reduce it enough that the probability of one moving from the intracellular to the extracellular is actually the same as the probability of one moving from the extracellular to the intracellular. Basically, the probability of you moving from the intracellular to the extracellular is still bigger than the probability of moving from the extracellular to the intracellular, even though the electrical potential difference is negative 65. You'd have to take it down to around minus 90 millivolts for the probabilities to actually become equal. Okay, so you still get a net movement of potassium out of the cell. Right, now if this is an electrical equilibrium, then that means that you can have no net movement of charge across this membrane. So this sodium current must equal this potassium current, because you're moving positive charge in the form of sodium, and you're moving positive charge out in the form of potassium. If you've got an electrical equilibrium, they are equal. So usually at resting membrane potential, these are equal basically. Now, of course, the ionic movements aren't equal. You've still got a net movement of sodium in and a net movement of potassium out, but the solution to that is just to put a pump in, which then moves the sodium out and moves the potassium back in, and that's what the sodium-potassium pump is, basically. Now, if you increase the potassium, if you add in a new potassium current out of the cell, then what you've effectively done is distorted this equilibrium. It was in nice equilibrium, but you've now added in another current into this picture, basically. So now you've made you're moving potassium ions out. So you've distorted this equilibrium, so you're going to now be moving more positive charge out of the cell than you are moving positive charge in, because this movement of sodium is in balance with the movement of potassium initially, but now you've increased the movement of potassium, so it's no longer in equilibrium, basically. So we've put in a new channel, this BK channel, and we've now got a bigger net movement of potassium out of the cell, basically. So this is BK. Okay, so that's going to cause, that's going to destroy the electrical equilibrium. So what are we doing? We're moving positive charge out of the cell. So that's moving positive charge from here to here. So we're going to make the outside more positive and the inside more negative. So that's going to make this electrical potential difference across the cell more negative, basically. So you're going to hyperpolarize the cell. So this turning on this BK channel leads to hyperpolarization. Okay, now if this axon terminal membrane is hyperpolarized, then it's less likely that action potentials will actually induce, that action potentials coming along the axon will actually induce action potentials in the axon membrane. Let me explain why. Because remember, for the axon, for action potentials in the axon membrane to lead to action potentials in the axon terminal membrane, what has to happen? Well, this portion of the membrane goes through action potential. It opens its voltage-gated sodium channels here. So it opens its voltage-gated sodium channels, which I'll just draw in full. So it opens its voltage-gated sodium channels. That lets sodium in and depolarizes this portion of the membrane to about plus 40 millivolts. I'm sorry, I've drawn the sodium outside. Yeah, sorry, sodium in there. So it depolarizes this membrane to plus 40 millivolts. Now what happens is that the sodium diffuses to the neighboring portions of the membrane, to these portions of membrane which are in the axon terminal, and it has to cause depolarization of this membrane up to threshold potential in order to activate the voltage-gated sodium channels over here. So basically, action potentials here cause action potentials over here because positive charge moves into the cell and then diffuses to the neighboring portions of membrane, which then causes the electrical potential difference across the membrane to rise up to threshold potential for the opening of the voltage-gated sodium channels. However, if your membrane is hyperpolarized, i.e. it's much lower than it usually is, so let me draw one of these graphs. Okay, so usually we're at about minus 65 millivolts. And to activate the voltage-gated sodium channels, you have to go to around minus 40 millivolts. So usually you can depolarize up to minus 40 millivolts. And that then activates the voltage-gated sodium channels and we're away, basically. However, if you move it down to, let's say, more negative potential, so let's say minus 80 millivolts maybe, then you are going to have to produce a much bigger initial depolarization to get you up to that minus 40 millivolts, basically. So you're going to need more ions moving from this neighboring portion into your axon terminal membrane to actually cause depolarization up to minus 40 millivolts and then cause an action potential to fire. So it's going to be less, so basically action potentials here are much less likely to induce action potentials here if this BK channel is active, basically. And that means that this, it means that this axon terminal is going to fire, is going to conduct action potentials far less, and that will stop the voltage-gated calcium channels becoming active, and then you'll stop building up more calcium. So you have then got an opportunity to pump the calcium that has come in out and restore a nice calcium level within your cell. Okay, so that's all for this video. In the next video, we'll discuss some interesting specific examples of the role of KCA1.